on individual B cells. The light chain and heavy chain genes coding for variable fragments of antibody in each cell are separately amplified by RT-PCR and then combined with the sequences by an overlapping PCR technique. For each cell, the Ig heavy and corresponding light chain gene transcripts are amplified by nested PCR and cloned into a mammalian expression system to produce human monoclonals with defined specificity in vitro. Using this approach to clone and express human mAb is unbiased, highly efficient, and requires only small cell numbers, which is a very big advantage considering making human monoclonals. The recombinant antibodies allow for direct conclusions on the frequency of specific human B cells in a diverse repertoire and may be able to rapidly generate numerous antibody-specific human monoclonals in a very short period of time. So recently, there is an improvisation to this technique where novel linear Ig, heavy and light chain gene expression cassettes are readily available now. And what do these cassettes do? Basically, these cassettes contain all the essential elements for transcriptional and translational regulation of the gene, including the CMV promoter, Ig leader sequences, constant regions of IgG1 heavy or Ig light chain, poly A tail, and substitutable variable and heavy and light chain genes. Advances in mAb expression cassettes constitute a highly efficient strategy for rapid expression of Ig genes for high throughput screening and analysis without cloning. So, regardless of the exact path that gives rise to the next generation of monoclonal antibody therapies, one thing is for certain. Therapeutic antibodies can do things that few small molecules can, and as such will remain firmly a part of the drug development landscape for years to come. To summarize, through the presentation in the last 45 minutes or so, I had touched upon hybridoma technology in brief and taken you through a small effort from our side on human monoclonal antibody as immunotherapeutic for COVID-19. We also discussed engineered human antibodies and how to develop fully human monoclonal antibodies. Thank you all for listening.
Dr. Sujata? Is Dr. Sujata there to moderate the questions?
Yeah, thank you, Dr. Madhur.
Yeah.
I'm there. I'm very much there. First of all, splendid and interesting talk. Madam, your talk was very, very lucid and interesting. I mean, you talked about various facets of development of human monoclonal antibodies in a very, very simple as well as very interesting manner for the benefit of audience, so that everybody could understand. I mean, right from the experienced seniors as well as the budding pharmacy students. So it was for all. So thank you very much, and best wishes and heartiest congratulations from our side for your noble venture. With your permission, can we take a few questions, madam? Sure. Yes, so there are very couple of interesting questions. Yeah, before I go to the chat screen, let me quickly summarize what you covered in your talk. You talked about, you first started off with the origin of monoclonal antibodies. You talked about brief history. Then you took over us to the journey of hybridoma technology to generate monoclonal antibodies. You talked about the pros and cons of passive immunotherapy. And further, you went ahead with the process of generating human monoclonal antibody using EBV transformation and various processes of making fully human therapeutic antibodies. So thank you so much once again. And I'll begin with a few questions. There's one question from Dr. Ashwini Kumar. And question from Dr. Ashwini Kumar is, what factors decide the formation of non-neutralizing and neutralizing antibody? Over to you,
Okay.
madam. Yeah.
Shall I answer this?
Yeah, ma'am.
Okay. So basically, what makes an antibody grossly neutralizing or non-neutralizing depends on whether it prevents a viral entry into the cell. So each virus gains entry into the host cell by means of certain receptors on the host cell. So if the viral antibodies to the viral protein is binding to a site which is of no consequence to the viral binding to the receptor, then entry into the entry into the host cell cannot be prevented. So then it does not neutralize the virus. So a neutralization activity happens only if you are able to prevent re-entry of the virus into the host cell. So we need antibodies which will stop the viral entry. So that is what distinguishes a neutralizing and a non-neutralizing antibody. So there could be many antibodies with very high affinities and titers to nucleocapsid protein. I had just mentioned about the Nature Medicine paper, where out of several clones, they had come up with about 15-20 antibodies which were able to bind with high affinity to the viral protein. Several antibodies made to the nucleocapsid protein was not able to neutralize the virus because that has no meaning at the level of re-entry of the virus into the host cell. So this is, did I answer your question?
Yeah, I think so. We have another question from Dr. Ashwini Kumar. Her question is, the second question, which is a continuation for the next, is how does HTRT, just give me a sec. Does EBV transformation use HTRT gene or any other method?
The transformation basically happens onto B cells which have C3b receptor. So regarding the gene that it utilizes, I will have to look up again. But all I know is it includes the C3b receptor on the B cells. So any B cell which has this C3b protein is able to bind to the EBV. So regarding the gene, I don't think I have the answer right now.
Thank you, ma'am. Thank you so much, ma'am. There's a question from Dr. Rahul Maheshwari from NMIMS Hyderabad. His question is, to increase the human content of therapeutic antibodies while preserving the specificity and affinity of parental non-human antibody is a major challenge. Is there any way to overcome this issue?
To retain the specificity, and also what is the second part?
Affinity of the parental non-human antibody, which is a major challenge. So is there any way to overcome this issue?
To retain, see, when we spoke of, I think the chimeric antibody and the reshaped antibodies in human antibody engineering. So what people have done is to keep the specificity of the non-human counterpart as close to it as possible. See, and then try to work around the framework region. They have done point mutations around the framework region to increase the CDR specificity and affinity. I did probably mention this point, maybe I was too fast. So what happens is, if you want to retain the specificity and affinity of the murine or a rodent antibody, at the same time, you want to minimize the murine component, you can do point mutations, because what happens in and around the framework region, if you can work around, you can increase affinities of the CDRs, which would have probably been compromised because of the grafting. Yeah.
Thank you, madam. There's
Hello. Sorry to interrupt, but Rajeshwari ma'am and Dr. Sujata, could you please put your video on
sure.
so that we can take some snaps also? Yeah.
Okay, fine.
So I'm just opening this, just a moment. Actually, my screen has not come back to normal after I quit my presentation. I'm just trying to figure it out. Just a second. I have actually totally lost the overall this thing of the Zoom, but I'm able to see Dr. Sujata. But
yeah, we
let me
can
just
see
check.
you now. I mean, we can surely see you. Yeah.
Can I
So you're
continue?
able to see me?
Yeah.
Probably. Yeah. You continue.
Can I continue, ma'am? Thank you, ma'am. We have a question from Dr. Vandana Patravale, our Vice President of CRS-IC. She has appreciated your talk, and she would like to know that what are the characterization tools one should have in-house to initiate research in this area?
Which area? COVID-19 or human maps?
Human maps, I understand.
Okay. What is the infrastructure or resources? What do you want to know? Tools. Okay.
Basic characterization tools or basic facilities that you could have in-house. I mean, if we need to initiate research in this area.
Okay, there are two components to the development of human antibodies. If you're talking of human map platform as such, of course, a cell culture room is a must. You need to have cell culture facilities. You need to have, if you're going to look at humanizing antibodies, you need to have an animal house facility to be able to inject animals. And we need to have a good molecular biology platform as well if you're going to do grafting, etc., etc. These are three main components which would be required for setting up any monoclonal antibody platform, whether it is murine or human. We would like to have these tools. And immunoassays and all that, I think, is routinely done in all the labs, so it shouldn't be a problem.
Thank you, ma'am. If you allow, we can take two more questions, ma'am. Can we? Just last two questions. Yeah. So we have Mr. Sachin Tandankar, and he would like to know what are the characterization parameters for antibodies.
An antibody, basically, first and foremost, any antibody is useful if it is highly specific and highly sensitive. And if it is an antibody made in vitro, we would say the clone should be stable. I always talk to my team in terms of 3S: stability, specificity, and sensitivity. Whether you talk of diagnostics or therapeutics, the 3S are very important. So I think that would answer his question, I guess.
That 3S is a very apt abbreviation which you mentioned. I mean, it's very apt in this context. Last question from Dr. Vikram Joshi.
Hello, sorry to interrupt, Dr. Sujata. We are a bit running late with our schedule.
Okay.
So we could probably take all these questions and we'll direct them to Dr. Rajeshwari. And Dr. Rajeshwari, if you don't mind, you could answer them on the email so that we can send it across to our participants later on. Yeah. Thank you.
Sure. I'll be happy to do.
Okay. So thank you so much, madam, for your talk. And as a token of our appreciation,
thank you so much.
Thank you so much once again. As a token of our appreciation, we would like to felicitate you with the certificate. So can I have Mr. Tarun, please? So the certificate rightly says, go green to save the planet. The reason why we mention this
thank you so much.
Because we at CRS-IC, we take conscious efforts with respect to environment. And the reason why we are mentioning this on the certificate is that as a token of our appreciation and as a memento, we will be planting a tree in your honor at Indira College of Pharmacy, Pune. So to commemorate this event and your significant contribution towards this workshop, CRS-IC will felicitate you by planting a tree in your name. So, of course, this is a virtual platform. We cannot thank you enough and clap, but thank you so much once again. Thank you very much, ma'am.
It's a real honor to me. Thank you.
Thank you very much. This is something which we are introducing it for the first time, planting a tree. So you are the first recipient of this kind of memento.
Oh, that's wonderful. Thank you so much.
Thank you very much. Over to, thank you, ma'am. Over to Dr. John, and he'll be, our next session will be by Dr. Suneet Shukla. So may I now request Dr. John to take over.
Good evening, everyone. Am I audible?
Yes, Dr. John.
Yeah. So welcome you all once again for this Control Release Society's Indian chapter uh, national level workshop on manufacturing characterization applications of monoclonal antibodies. Our next speaker is Dr. Suneet Shukla, who is a pharmacist with PhD in molecular pharmacology from Jawaharlal Nehru University, New Delhi, India. He has completed his postdoctoral training in National Cancer Institute NIH. He then joined FDA and has worked at the Office of Pharmaceutical Quality and Office of Generic Drugs, where his responsibilities included the assessment of INDs, NDAs, and DAs. Currently, he is working as senior pharmacologist at the Office of Clinical Pharmacology, USFDA. Dr. Shukla possesses years of highly specialized research and regulatory experience in PKPD data, analysis, in vitro drug release and dissolution analysis, bioequivalence determination, IVIVC model development, and validation of SUPAC changes or bioweber applications, quantitative modeling, and simulation analysis for small and large molecules from early to late drug development, including drug-drug, drug-excipient interactions using transport-mediated IVE PBPK models to understand drug absorption and oral bioavailability. So on the behalf of CRS-IC, I heartily welcome you, sir, and request you to kindly proceed with your presentation, please.
Thank you. Thank you for your kind introduction. Can you all hear me?
Yes. Yes,
Okay.
you're audible.
Okay, let me start my screen share for a second.
Dr. Pete, can you please allow him to share his screen?
Yeah, I think I can. I'm just trying to figure out
Okay. Yeah, yeah.
if I share my screen.
Yes. You can go for the slideshow now.
Yeah. Can you see the slideshow or you see?
No.
Okay. A
Yes.
second.
Now it is there. Yeah.
Is it the slideshow now? You can see
No,
slideshow?
no, but the slide is not properly visible.
Okay, but hold on. One more thing. Let me just see because my Zoom.
Instead of using, because it is becoming very small icon, no, so you can try taking the main menus upside.
Yeah. Hold on one more time.
Yeah. In the slideshow you can go for, yes,
Can you see
yes,
my slideshow or the real
you
slides?
can click that, please. It's
I
not
did that because I have multiple monitors, so looks like
okay.
it's not working. I think I'll go ahead with this view. Probably you can see the slides, right?
Yes, yes, yes.
Okay. So thank you for the organizers of IRS Indian chapter, CRS sorry Indian chapter, for invitation, and Dr. D'Souza for your kind introduction. And congratulations to you all for launching this new website. I'm sure that this will help to promote CRS activities and events to the broader community. And I wish you all the best for your website and all of the activities. So today I'm going to talk about the basics of monoclonal antibodies drug development. And if I go back to my disclaimers, I work for the FDA, so the remarks which are presented today are my own personal views, and they do not represent those of FDA or the US government and should not be construed as FDA policy or advice. Here's a brief outline of my presentation. it would be introduction and followed by hold on a sec. So it'll be introduction followed by the monoclonal antibody introduction, followed by some aspects of preclinical and clinical development, and also clinical pharmacology considerations, which are important for developing biologics and monoclonal antibodies, followed by some concepts about biosimilarity. I intentionally included biosimilars in this talk as there is an increasing interest in the development of biosimilar from Indian pharmaceutical industry, and I think that's the way forward. And as Dr. Ajit Singh mentioned earlier too, we are considered good in terms of the generic drug industry, but now that biosimilar is the new era, and the Indian pharmaceutical industry has to catch up with that, and that's where biosimilars is one important area where a lot of things are going on. I also understand that there are many undergraduate and graduate students who are attending the workshop today. Therefore, I'm going to present basics of monoclonal or biosimilar development. And because I'm a pharmacologist, I would try to put my emphasis on the pharmacology and clinical pharmacology aspects of these drug development. Can you still see my screen? I'm not sure.
Yes, we can.
Okay, good. Thank you. So biological products are ones which are regulated by FDA, and they are used to in the diagnosis, prevention, and treatment and cure of diseases as defined by definition of drugs by US CFR. And these products are from a diverse category of products which are generally large and complex molecules. These products may be produced through biotechnology in a living system, such as microorganism, plant cell, or animal cell. And Dr. Rajeshwari covered quite a bit of monoclonal development antibodies in animal cells and how it is done. So thank you for the background. There are many types of biological products which are approved for use in US, including therapeutic proteins such as pegfilgrastim, monoclonal antibodies such as adalimumab, and vaccines such as those for influenza and tetanus. So in terms of the biologic regulatory history, it goes back almost 100 plus years. This is like brief history of what has been done by FDA over last 100 plus years. It started with the Biologic Control Act in 1902, which was enacted in response to two incidents involving the deaths of 22 children who had contracted tetanus from contaminated and from there onwards, in early 1944 and 38, we had this Public Health Safety Act, which was enacted, and then the Food, Drug, and Cosmetics Act, which was enacted. Both had emphasis on biologics. I would say in early 70s and 80s, when the FDA Bureau of Biologics was established, followed by FDA Center for Drugs and Biologics, that's where a lot more activity happened. In 1987, FDA was sort of separated into two separate centers, Center for Biologics and Center for Drugs. And then in 2003, the therapeutic biologics moved to the Center for Drugs, where I work, Center for Drugs Evaluation and Research. In terms of the monoclonal antibodies and biologics in general, I would say last 10 years have been a big area where a big period where a lot has been done. In 2010.
Interrupt you, doctor, but we can see slide number 4.
Oh, okay. I'm so sorry. Can you see my slide now?
100 plus years?
Yes.
Okay.
Okay. I'm sorry. I think I have multiple monitors, so Zoom is not syncing with the other monitor. I'll try to move both monitors then. Yeah, so I just go back to what I was telling. I said that there is 100+ years of history for biologics approval by FDA, and in last 10 years, I would say the majority of the work in the approvals of biologics has been done. We had in 2012 we had the Biosimilar User Fee Act, which was enacted, which came because of the BPCI Act, which was initiated in 2010. Then in between 2015 and 2019, we had the CMC guidance for biologics. 2017, we had a Clean Farm guidance. And 2019, we also had guidance on immunogenicity and with interchangeability guidance also in the same year. So I would say a lot has happened in the last 10 years for the development of biologics. And as you can see at the bottom of the slide, and as Dr. Rajeshwari also mentioned, the first monoclonal antibody which was approved by FDA was in 1986. Before that, the biologics in terms of genetically engineered biologics was insulin, which was approved in 1980. And recently, we have approved many more monoclonal antibodies going from 2010 to 2015, and now we have many more biosimilars also, which are approved by the FDA. Now, just for the students, to put things in perspective, there is a difference between monoclonal antibody and a small molecule, as you say, between a large molecule and a small molecule. And this is shown by the size here. Single monoclonal antibodies weighs more than 800 times than an aspirin molecule, which is shown here on the left, which is a very small molecule. So in terms of the clinical and non-clinical characterization, the actual set of assays and the aspects which are considered are different because these two molecules are very different too. And also in terms of the differences, as Dr. Jising mentioned about the generic industry, which is already very established, which is focused on the small molecule drugs. As I mentioned in the previous slide, the size is a big difference. Also, the manufacturing process for small molecules, it is based on chemical synthesis, which use a predictable chemical process. You can make identical copy of the medication readily. And after the chemical is synthesized, it's stable. And also an example would be aspirin or any small molecule or corticosteroid drugs are example of small molecule drugs. And that's where the generic industry is very well established. On the other side, for large molecule drugs, which includes all the biologics, the size is a big, huge difference. On top of that, because they are produced in the living cells under culture, they are actually grown. So the manufacturing process is complicated and hard to control. And identical copy of medication is not possible, although it is close to, especially in the terms of biosimilars, it is close to the reference drug in there. Stability is a big concern. Unstable due to the sensitivity and external conditions is a big problem with biologics. Example is Humira and Botox. I think a couple of days back, there was a news about the coronavirus vaccines, and one of those vaccines is supposed to be stable under -70 to -90 degrees centigrade. So, and that comes from the unstable aspect of that biologic. And so you can imagine if a vaccine has to be stored at between -70 and -19, it would be a logistical nightmare for that vaccine to be distributed through the pharmacy chains and the hospitals. So there are logistical challenges associated with large molecule drugs, associated with the development and also through the distribution. So moving on to the next topic, which is monoclonal antibodies. I think Dr. Rajeshwari provided a nice overview about the structure of the monoclonal antibody. As I just would like to show that there is this antigen, which is a toxin or other foreign substance, which induces an immune response in the body, especially for the production of antibody. And this antigen has an epitope, which is a blue part here, which shows the part which binds to the paratope on the antibody. So antibody actually is a whole molecule, is a blood protein producing response to and counteracting a specific antigen. The FAB part, which is shown on the circle, is the part which is fragment antigen binding region. It binds to the antibody, it binds to the antigen, and the FC part, which is the bottom part, which is a crystallizable region in the tail region of the antibody. It interacts with the cell surface receptors called FC receptors, and also some proteins of the complement system. Now, there are heavy chain and a light chain associated with this structure, which is also shown on the figure here. Epitope is a part of the antigen molecule to which an antibody attaches, and paratope is a part of an antibody which recognizes and binds to the antigen molecule. So in terms of the classification of human immunoglobulins and the type, these heavy chains are designated by the Greek letters alpha, gamma, delta, epsilon, and micron. And the immunoglobulins are produced, that's why they are labeled as IgG, A, G, D, and M respectively. And also from the light chain perspective, it could be either of a kappa subtype or a lambda chain subtype. And if you look, and that's how the antibody is classified. For example, belimumab is a human IgG, one with a lambda light chain, and golimumab is an IgG, one with a kappa side light chain in there. So this is how you put a nomenclature to the antibodies. Um, In terms of the binding of antibody to the receptor, as I mentioned in the structure, the FAB region is the one which identifies antigen to the cell surface, and the FC region is the one which is a crystallizable region which binds with which accounts for the non-specific binding. So there are two types of binding: the specific binding and the non-specific binding. Non-specific is through the FC region, and the specific is through the FAB regions. And the good thing about the FAB and antigen interaction is it's almost irreversible kind of a binding because of a very high affinity of the antigen antibody interaction, which goes almost into to picomolar affinity. Now, coming to the preclinical and the clinical aspects of antibody drug development, what I am showing here are the different stages of target validation, different stages of the preclinical antibody drug development. And these stages are associated with different objectives. And for achieving these objectives, you have to do several different types of studies. So first stage of our preclinical antibody drug development, as Dr. Rajeshwari also mentioned, is target validation and molecule selection. So in that stage, you have to identify the objective would be to identify pharmacologically relevant species for efficacy, safety, and demonstrating the PK, establish the mechanism of action, and identify biomarkers, and with the efficacy endpoint, and how you have to evaluate if the molecule is good enough to go in terms of the PK aspects from development perspective. And in order to achieve that objective, there are several assays such as affinity and specificity characterization. As I mentioned in the previous slide, the antigen antibody interaction needs to be very specific, and that's what is done at the preclinical stage. In vitro and in silico screening is also done for the lead molecule, and assessing if it is possible to develop that, and preliminary assay strategies for measuring all these, achieving all these objectives are identified through these studies. Also, when you come to the next stage, which is basically lead optimization, the objectives change a little bit. Now you have a target, a molecule which is in your hand, and a lot of it is similar to small molecule field where you characterize the PK and initiate PK/PD studies in if, efficacy and safety models, establish exposure and response relationship, and the objective would be to identify if there is early PK liability, which can be done using a rodent model. Also, dose ranging studies, which is very important in the non-clinical species, and that should be done in a species which is more closely related to the target, which is present in human population too. And then followed by pilot PKPD studies and safety studies and preliminary modeling to come up to a point where you have an idea about the dose which would be used for human. That brings to the next stage in the preclinical development, which is IND enabling studies. And I would say that's where the work start in terms of putting things for going into first in human trials. That's the objective would at that stage where you are ready for doing the IND is to integrate pharmacology and toxicology data to prepare the IND package for starting for entering in the human dose. You define the maximum tolerable dose, you select the first in human dose, you project efficacious dose in human, and also design dose escalation scheme. Those are the objectives. And a lot of that is done based on the toxicokinetic data, which is already available to evaluate the safety. And then the PK in non-human primate and its projection in the human PK in the human population is important because that's how you will come to your first in human dose.
Dr. Sunit,
Yes.
I'm sorry to interrupt you in between. Can you just share the screen where you have a full screen view of this slide? Because some people who are viewing on mobile
Uh-huh.
having
Okay.
difficulty seeing the slide. So you just on the share screen, you select the
Yeah.
your display which has a full screen mode of your slide,
Okay. I am trying to let me try one more.
because you have display screen 1, 2, 3, 4, whatever, how many
Yes.
screens you
Yeah. Yeah.
yeah. So just share other screen, instead of display, just change the display screen number, and I'll tell you, which one is correct.
Okay. Let me see.
No, not this one. This is showing your browser.
Okay.
Just press F5 here.
Hold on. How about this now?
Yeah, this correct. This correct. Perfect.
Okay. Okay. Good. Um, Yeah, so in terms of stage where you are at the IND, you need to make sure that the toxicokinetic data which you have supports the first in human dose, and that's when you would go into the clinical development. And a lot of this data which has been acquired until this time, which is all preclinical, would be used to justify the clinical development. So in terms of the clinical development, it's more or less same as what you would do in for a small molecule. When you file an IND, typically it's a 30-day safety review where mostly in vivo gene tox, safety pharmacology, preliminary PK data, which you have gathered through the non-tox studies, gene tox data, repeat tox, those studies, all of that is part of the non-tox studies which have been done that is used to file the IND. And then it goes through phase 1, phase 2, and phase 3 trials as you would do typically for a small molecule change, which is associated with further physicochemical characterization in terms of CMC, if there is a process or a formulation change or even a dosage form change that needs to be considered through these phases of trials, and a bridging needs to be done from pharmacology perspective, because let's say if at the IND stage you started with a formulation which was a subcutaneous formulation and an IV formulation, and at the phase 2 stage, because of the commercial and the logistic reasons, you need to move to a different kind of formulation. So from PK perspective, a bridging study needs to be done. And also from the repeat dose perspective, because a lot of these monoclonal antibodies specifically are given for a long duration of time, all that data needs to be supported by repeat dose studies also. So at the end of the phase 3 studies, you file your BLA or NDA as the case may be, and associated that's where carcinogenicity or data also is very important. And what we have seen in terms of biologics specifically, a lot of data is considered as a post-marketing requirement or commitment after the NDA is approved. That brings me to the next section, which is the clinical pharmacology considerations for biologics per se. And this, as I mentioned previously, that the maximum safe starting dose would be something which is very important in the context of initiating first in human studies, that too in human healthy volunteers. And the FDA has a guidance which is now on almost 15 years, going back 15 years old, where we use the principle of the no observed adverse effect level, and for biologics, on top of that, the minimal anticipated biological effect level. These two uh, these two aspects are considered to determine the first in human dose, and we'll discuss individually these two aspects. I would go back to I think back in 2006, there was a study which was done for anti-CD28 molecule. T-Genero was the company which was developing this in UK. They had very good, robust non-clinical data for their biologic molecule. But when they initiated the first phase one in human studies with 6 subjects, they observed a lot of cytokine storm, and there was organ failure in those 6 subjects. That's where, so before that, the concept of no observed adverse effect level used to be in place. We used to follow that all the time. But after that, there is this new concept, which is minimal anticipated biological effect level came into picture, especially in the for the case of biologics. So how does it work? So basically, you from the preclinical data, you identify no observed adverse event level, NOEL level, which where you gave a range of doses to animals, respectively 3 different animal species, and you identify a dose where you don't observe any adverse event. And that adverse event, once, and then also on the pharmacological assessment side, you identify uh, a dose where you have the minimal anticipated biological effect. It could be through an in vitro or an in vivo assay observed. So once the NOEL and the MABEL doses have been determined from toxicology and pharmacology studies, they are considered together in the selection of the maximal recommended starting dose as shown in this slide here. So the left-hand panel, it determines it describes the assessment of the toxicology and the conversion of the NOEL to a human equivalent dose. And that goes by these steps. And these steps are in the guidance, which I showed previously. So for monoclonal antibodies that have a molecular weight of more than 100 kilodalton, which is basically full length antibodies, and they are being administered through IV, where the distribution of the drug is assumed to be largely confined to the vascular space, the human equivalent dose is based on the milligram per kilogram. So once you have the NOEL, you convert into human equivalent dose. And that is conversion is basic in allometric scaling, where you identify that dose which is good for human population. And then you apply this tenfold safety factor for the NOEL, and that would be your dose. And we'll go to the example a couple of slides later. Again, individual numbers, couple of slides again. On the other hand, the right panel describes the assessment of the pharmacology and determination of MABEL. And that is important for the consideration of the pharmaceutically active dose. So basically, MABEL is the minimum anticipated biological effect level. You identify a concentration where you start to get that biological effect. And that is done through an in vitro assay and then integrating the data into a PKPD modeling approach if it's possible. And then that approach accounts for animal to human differences in affinity versus potency, also animal to human differences in exposure and distribution, and what would be the anticipated duration. So once you have the Noel and Mabel calculated this way, the maximum recommended safe starting dose, which is MRIST, as I mentioned earlier, is the one which you would use, which is basically small, the smaller of these two doses. And we'll discuss, we'll look into the numbers in the following slide after this one. So the determination of this safety margin, this is very important, especially for biologics. It's very important. The Noel studies should be conducted in clinically relevant species, and the safety margins are calculated using multiples of the human dose, which is relative to the PK exposure data. And the safety margins, they vary between product class or clinical indication, and determining the safe starting dose and dose escalation for clinical trial, the safety margin is very important. Going back to the slide which I presented again, and this is coming from the publication, I'm giving you an example. Like if you identify a Noel in monkey and rat, which is 30 milligram per kgs and 100 milligram per kgs. So directly, because if you can convert to milligram per kg, it's basically the same dose, which is 30 milligram kgs and 100 milligram per kgs. And now if you look between these two doses, 30 is obviously lower. So that would be the Noel, especially if it's from monkeys, so that's more reliable. And then you apply a 10-fold safety factor, which brings it to 3 milligrams per kg. So that would be an ideal starting dose. If it was it's a small molecule world, probably we would start with this dose. But in the case of biologics, this additional step, which was incorporated sort of a strategy after 2006, the concept of MABEL, where the monoclonal antibody is known. Now we are going into the mechanism of this antibody. It's a hypothetical antibody which is known agonist for to induce the depletion of lymphocyte subset. For that, a dose of 0.1 milligram per kg is achieved. At this dose, 10% of receptor saturation of target lymphocyte receptor in monkeys is observed, and that leads to the cell depletion of less than 10%. So this is the data which we have for the pharmacology aspect now in terms of the assay. So considering that there may be a 5-fold lower potency in monkey versus human, the dose of 0.1 milligram per kg would be divided by a factor of 5, which leads to 0.02 milligram per kg. And that would be the MABEL dose. Now, if you look at the Noel dose and MABEL dose, there's a difference. Because of the biologics, we would go by a lower number. That's why the maximum recommended starting dose would be 0.02 milligram per kg. That's based on the MABEL. So those are the general principles for identifying starting dose before you go from a non-clinical to first in human studies. So MABEL actually is a useful number for protein drugs because it defines a dose at which receptor occupancy is low. And taken together, the first in human doses need to be calculated from both Noel and MABEL, and the lowest value is recommended for the clinical trial. Generally speaking, monoclonal antibodies with agonistic modes of action, they require no more than 10% receptor occupancy, and that would be the proposed starting dose. If it's an antagonistic mode of action, they may need to have a higher dose, but that depends on the indication as well. So I just try to give you a flavor of how you arrive to the phase 1 first in human starting dose study. And then you go to the dose selection for phase 2 and phase 3, which is basically following the same path as the small molecule world. You have dose ranging studies. The dose needs to be identified. It should be a linear PK versus a non-linear PK. Depends on if it's a in the case of linear PK, it's mainly attributable to the Fc receptor mediated clearance. For a nonlinear PK, it's mediated through the targeted mediated drug solution, and then you have to consider target occupancy, exposure response relationship, and dosing intervals. Those are the factors you would consider for your normal small molecule drug development as well. QT assessment is not important for monoclonal antibodies, as they have very low potential to block the human ERG channels, and these ERG channels are responsible for prolonging the QT intervals. So generally speaking, TQT studies, the target the the, the the studies are not required for monoclonal antibodies unless there is a specific signals, but routine ECG monitoring in phase 1, 2, and 3 clinical studies are done. There is a possible interaction of drug-drug interaction between biologics and small molecules, and that also is considered because there can be two different types of interactions. Biologics can interact with small molecules, or small molecules can have an effect on biologics activity. In terms of biologics and small molecules, they can modulate the drug metabolizing enzymes, and also they can have an effect on transporters such as PGP, and that's how they would alter the pharmacology of small molecules. And the effect of small molecule and biologics would be a decreased immunogenicity. And one of that principle is used and in a lot of these therapies where methotrexate is used to sort of act as an immunosuppressant with biologics, especially in the case of rheumatoid arthritis. In terms of special population consideration, because monoclonal antibodies which are higher than 70 kilodalton, renal excretion is not a big concern. So therefore, generally, renal impaired patients are not required. Also, biologics are not eliminated by liver, so hepatic impairment is also not required for the assessment of the impairment. Dedicated PK study in hepatic impaired patient is generally not required, but population PK can be used to verify the dose. Immunogenicity, as touched by Dr. is a concern. The formation of the neutralizing anti-drug antibodies against the corresponding biologic or immune complexes, which trigger the reticuloendothelial system, results in increased clearance of a biologic, and that leads to a decreased C drop concentration if the immunogenicity is increasing. The degree of immunization, as Dr. Rajeshwari also mentioned, route of administration, duration of therapy, that can all impact the immunogenicity. And the incidence of immunogenicity is negatively correlated with the degree of immunization of monoclonal antibody. How much time do I have here?
Yeah, you can proceed, sir, for another 10 minutes.
Okay. So this
10 to
is my
15
last
minutes.
topic about biosimilarity, and as I mentioned,
Time, sir. Please.
okay, as I mentioned during my introduction, this is an important part of the biologics development. Biosimilar and interchangeable drugs are like generics in large molecules. And the biologics came into picture with the enactment of BPCI Act, which was passed in 2009. As a 2009, it was 2009 BPCI Act, which was passed in 2010, and it created an abbreviated licensure pathway for biological products, which are shown to be biosimilar or interchangeable with an FDA licensed substance product. So it's a generic version of a biologic molecule. So these are two separate concepts here: biosimilar and interchangeability. And I would like to just emphasize that these are two different concepts, because sometimes we have seen people using these two terms interchangeable. It's not the case. Biosimilar drugs are highly similar to the reference product, notwithstanding minor differences in the clinically active components. And the premise is that there's no clinical meaningful differences between the biological product and the reference product in terms of safety, purity, and potency. While interchangeable product is still a biosimilar product, which is to the reference product, but it can be expected to produce the same clinical result as a reference product. That's also true for biosimilar. And this is an important concept here that for a biological product that is administered more than once to an individual, the risk in terms of safety and diminished efficacy of alternating or switching between the use of biological product and the reference product is not greater than the risk of using reference product without an alternational switch. Just to put in a simple language, interchangeable products can be basically biosimilars that can be changed at the level of the pharmacist, where the pharmacist can make that determination and they can be basically switched on and off between biosimilar and interchangeable. But biosimilar products cannot be an interchangeable products. They can only be changed at the level of the physician. So under this abbreviated licensor pathway for biological product, the premise is that similar to what we do in the generic world, here the product needs to be highly similar to FDA licensed biological product, which is the reference product. And the licensor pathway is 351K, which needs less than a full complement of product specific preclinical and clinical data. That's why it's called abbreviated licensor pathway. And here is in terms of the principles which are used. So if you look on the left side, this is basically originator biologic 351A pathway, which is for the reference drug. It starts with molecular characterization, non-clinical studies, phase 1 clinical, 2 and 3, as I discussed earlier in my slides. So this is how the path goes in terms of the original biologic 351A path. But for biosimilars, this is sort of reversed, the pyramid is inverted. The basis of the approval for a biosimilar is the physicochemical characterization associated with biologic characterization. So the analytical characterization of the biosimilar product with the reference product is very, very important, and that forms the basis of the approval. And then after this is done, if there is a residual uncertainty, non-clinical studies, PKPD studies, and clinical studies, they all need to be submitted, but the basis is this. And the objective of this 351K path, which is a biosimilar path, is not to demonstrate the safety and efficacy, but to demonstrate the similarity. So in terms of the general requirements, it should be the under 351K application, a biosimilar should be biosimilar to the reference product, which utilizes the same mechanism of action. The conditions of use which are proposed in the labeling for biosimilar should have been the ones which have been previously approved for the reference product. They should have the same route of administration, dosage form and strength, and should meet the standards for the safety, purity, and potency as for any other biological product. Just to reiterate again, the goal of the standalone and biosimilar developmental programs are different. The goal of the standalone program is to demonstrate that the product is safe and efficacious, while where the drug development starts with preclinical, followed by phase 1 and 2 pivotal trials. Versus the goal of biosimilar is to demonstrate biosimilarity between the proposed product and the reference product here. Again, to emphasize, the goal is not to independently establish safety and effectiveness of the proposed product. So FDA has outlined the stepwise approach, which is given in the link below, where you basically look at the residual uncertainty at each step and the totality of evidence, which is important for evaluating the biosimilarity. There's no one pivotal study that demonstrates biosimilarity. Instead, you apply a stepwise approach, starting with the analytical similarity and determine what is the residual uncertainty, what differences have been observed, and how best to evaluate the potential impact, and what studies will be needed to address the residual uncertainty. In terms of the PKPD studies, because I'm a pharmacologist, again, I would like to say that you start with a similar PK exposure and PD, which provides the same response. And the assumption is that you have the same exposure and same response. the biosimilar will have a similar efficacy and safety. And if it will also tell you if there is a clinically meaningful difference, then you go to the clinical study part of it, which is comparative clinical study. So clinical PK data should be expected, but it's basically comparative clinical study, and PD data is desirable, but and also based on the case by case considerations. Immunogenicity, immunogenicity, as I mentioned in our previous slides as well, it's very important and it should be collected at all clinical studies. In terms of the comparative clinical studies, if there are clinical meaningful differences between the proposed product and reference product, they should be investigated. And the goal again is to have the equivalence, which is basically demonstrating that your product is non-superior and non-inferior to the reference product. And study population can be same population as for the indicated product or different, but the same population may aid in the determination of treatment effects and selection of margin, while different population may be more sensitive to detecting differences because of the lack of background noise. But again, those decisions are made depending on the type of the biosimilar which you are studying. Biosimilars cannot be licensed for a condition of use that is different from the reference product. And that basically goes back to the principle of the generic drug development. And the PK assessment is followed in the comparative clinical studies, also equally important. That brings me to the summary of especially the biosimilar part. So demonstrating biosimilarity is different from the standalone product. The approach and the development program should and will be different based on the intended outcome to demonstrate biosimilarity. As I mentioned, analytical similarity data is the foundation of biosimilar development. Understanding the relationship between quality attributes and the clinical and safety efficacy profile aids ability to determine residual uncertainty about biosimilar and the predicted clinical similarity of the quality data. So the nature of the clinical studies, they will depend on the extent of the residual uncertainty about the biosimilarity of the two products after conducting an extensive analytical similarity assessment. These residual uncertainty need to be eliminated. So you can definitely say that there are no clinically meaningful differences between the proposed biosimilar and reference product. Scientific justification must be provided to support extrapolation to other conditions of use. And the content of a biosimilar development program is based on the stepwise development, and approvability is based on the totality of the evidence submitted by the sponsor. I think that is my last slide. Just want to emphasize that we are in this very exciting era of biosimilar development. What is shown here are the number of biosimilars for which the biologics for which the patent is expiring. And for quite a few of them, as you can see, the ones which are in red or yellow, the biosimilars are either being filed or approved because they are the patent for the reference drug has already expired, and for the new drugs which are and the green ones are the ones which are marketed, but there are quite a few for which the patents have not expired yet, but they are going to expire as is shown in the slide, and the field is open for biosimilars to be developed for these drugs. That is all I have to present. I would like to acknowledge my division members who provided background information and some of these slides as well. And thank you for your time, and I'm ready to take questions. Thanks.
Dr. John.
Thank you, Dr. Sunit Shukla. In fact, coordinator Dr. John D'Souza has lost the connectivity due to internet issues. So thank you so much for that presentation and taking us through the total route, the preclinical development specifically. So we'll take few questions from the chat box. There are one or two questions, if you're okay with it.
Yes, please. Yeah.
Okay, so there's a question from Sandhya Shenoy. She asks, is the FDA approval process for the biosimilar as rigorous to the reference? Are any changes to the labeling allowed?
No, I no. To my knowledge, especially from a clinical and clin farm perspective, in terms of the indication, you cannot have any changes compared to the reference listed drug. So if it's approved for one indication, and there is another indication for which you would like to extrapolate the data, that may be allowed depending on, again, the concept of residual uncertainty. And from a clin farm perspective, it's the sort of the similarity between the PK/PD exposure response relationship. If that is established, the extrapolation to the indications which are already described for the listed drug, although you have not done those studies, may be possible, but you cannot have a change in the label. It's basically, because it's an abbreviated path, it's going back to the same concept as the generic drug, where you have to have the same indication, same dose, and the same type of characterization.
Thank you so much. There's another one from Ashwini. And question is, if biosimilar is very similar to original biologic in structure as well as potency, what benefit will new class of bio-betters have?
Okay, so I mean, that's a question for maybe some CMC colleagues to answer. But what I can say in terms of the new class of biobetters, as long as you, and I think I mentioned in one of my slides, in terms of non-superior and non-inferior, we don't want to have a product which is either superior or inferior to the reference listed drug, because we don't have the safety and efficacy data associated with a similar, a different exposure response relationship. So again, from clin farm perspective, we would not be comfortable, or we would not know what happens if it's a biobetter, because we don't know what are the implications of having separate, having an improved sort of better outcome. So I would say the concept of the abbreviated path and the biosimilar is to have access to many more products which are same as reference product, but at the same time you have options, and that would eventually bring the price of and the accessibility of these medications down because of the competition.
So there are many other questions I can see in the chat box, but I'll just take one last one. Question is from Dr. Manjapa, who asks, where do FE bodies stand over MABs in preclinical or clinical development?
Okay, so that may be a question for Dr. Rajeshwari to answer. To my knowledge, I have not seen an antibody in clinical development yet, and I could be wrong. I have not seen all the antibodies yet, but mostly the ones which we have seen are these monoclonal antibodies, or like ADC type of complexes, even some FABs. Antibodies I have not yet come across, and also in terms of clinical characterization, I'm not aware about that.
Okay, thank you so much, Dr. Sunit Shukla, and over to Madhur for doing the honors on behalf of CRS-IC.
Thank you so much, Dr. Patravale, for asking questions on behalf of Dr. D'Souza, and thank you for the invitation as well.
It was our pleasure to have you on board, and we look forward to more such opportunities. So
yeah, it's an honor.
I think on the screen you can see that, go green to save the planet. And as mentioned to the earlier speaker, we would be planting a tree in your name, in fact, in Indira College of Pharmacy at Pune. So thank you once again for being with us this evening. In fact, of course, your morning. Thank you so much.
Thank you again for the invitation. I really enjoyed, and I'm looking forward to the next talk. Thanks again.
Our pleasure. Thank you so much. Dr. Pethe can take over.
Yes, ma'am. Dr. Sachin is there? Yeah. It's an honor and privilege to welcome the speaker, the last speaker for today's workshop, Dr. Sachin Dubey. Dr. Sachin Dubey is presently working with ICNO Sciences, Switzerland as Deputy Director and Head of the Formulation, Analytical and Drug Product Development. His current responsibilities include designing and executing product development and characterization strategies for both early and late stage product for biologics. Sachin earned his PhD from the University of Geneva, Switzerland, and has previously worked with Glenmark Pharmaceutical Switzerland and Novozymes Biopharma, Denmark. He has more than 12 years of experience in biopharmaceutical formulation and combinational product development. 12 products developed by him and his team are currently in different clinical trials. He has overseen the production of more than 75 drug product diluent for clinical trial phases from 1 to 3. He has extensive experience with regulatory submission in the US FDA, European Union, and India, and 9 PCT patent applications, more than 20 publications in very high impact factor journals, 6 book chapters, and more than 60 presentations to his credit. He also has received 20 research awards, including prestigious industry awards from Glenmark, Merck, Novartis, and Swiss Society of Pharmaceutical Sciences. With this brief introduction, now I hand over the session to Dr. Sachin for his talk on role of antibodies-based therapeutics in the modern healthcare system. Over to you, Dr. Sachin.
Great, thanks a lot. So let me check this. How is yours? Very good, very good. I would I would sincerely like to thank you. I mean, it's Saturday evening, 9 o'clock, and so many keen listener here. I can only salute you. So thank you, and thanks also for the kind word. And let's dive into the presentation here. Can you see the presentation?
Not yet. Yeah. No.
Yes, it's there, Dr. Sachin.
Perfect. Very good. Okay. I was keenly listening to the previous two talks, and maybe I can start from one of the question which was let's say handed over to CMC. Me being one of the member of CMC here in biopharma world, the BioBetter, the question of BioBetter here, and the question was why BioBetter versus biosimilar, or where do you place BioBetter compared to biosimilars and innovator products? The key differentiation, I should say, for BioBetter is that it employs the same target while the molecule itself can differ. And for instance, if the originator molecule have been developed 5, 7, 10 years back, the technology evolves, right? The original molecule may be poor on stability, may not have sufficient shelf life or even half-life in the body. And a BioBetter can be developed overcoming some of these challenges while keeping the target same. So that's where, from an industrial perspective, BioBetter will be placed, where you improve on the molecule itself while the target is still kept the same on a very high level. Okay, now coming into what I am supposed to talk. My main responsibility here today is that we have learned already how to produce, we have learned how to get it approved. Now, the thing is how to use them, how we use monoclonal antibodies and all the other associated derivatives of monoclonal antibodies over the next 40-45 minutes. Once again, quick glimpse on what we will look into. We'll discuss about therapeutic antibodies here. Then we'll look into some of the other aspects. I'm sorry. Yeah. Okay. It's better. Okay. Then I will look into what are some of the other applications of monoclonal antibodies still remaining into the therapeutic angle, and finally getting into some of the allied areas like purification. I think I can surely skip this slide. This have been gone over by both of the previous speakers, so I can really get over some of the initial slides. Really, I don't just repeating the information. That's on a very high level structure of a monoclonal antibody discussed over already previously. The only thing I would like to bring to your attention is that there are two things which are very important here. Obviously, the antigen binding region. So this is the target of the monoclonal antibody. And the second part, which is also important, or I would say not less important than the antigen binding region, is the effector function. And we'll dive into how, from an industrial perspective, we make use of the secondary or the effector function as well. Quickly going into what are the different types of IgGs, we have seen there are 5 different class of Igs. Within IgGs, there are different classes as well, starting from G1 to 4. And structurally, they are different. Structurally as well as functionally, they are quite different here as well. Just a summary of their properties, and the key differences when it comes to IgG 1 to 4 are highlighted here. The IgG, let me go with IgG 3 and 4, which are the least used in pharmaceutical industry, biopharmaceutical industry. And there are reasons to that. IgG 3 have only 7 days of half-life within the body, as compared to 21 days of half-life for all the other IgGs. So that makes it much less suitable. On top of that, the hinge region, which has a lot of cysteine bonds, which makes it much more flexible, or in other words, more difficult to stabilize compared to the other IgGs. IgG2 is the most of the targets for IgGs, whether it's IgG1, 4, is actually one of the receptor, one of the protein within the body. And if you see from this table, IgG2 is not always generating antibody response against protein. It's very good when an antibody response is required against polysaccharide, but for protein receptors, it's not the best selection. So in theory, we are left with IgG1 and IgG4. And among them, IgG1 is, in our body, its relative abundance is 60%. And on top of that, there are other properties which are listed on the bottom of the table here, which are one more time driven by the secondary function or receptor function, driven by crystallizable fraction of the molecule. While IgG1 have higher propensity for effector functions, IgG4 have, on a comparative level, lower propensity to engage complementary activation or other function, other secondary function. And I will explain you why these functions are important. And just keep it in mind that IgG1 elicit the secondary functions, while IgG4 lesser than IgG1. Now, just quickly having a look on what are these effector functions on a very high level one more time. Um, these are nothing but how our immune system works, like ADCC, antibody dependent cell mediated cytotoxicity. You have a cell which has to be taken out, and then there are several of killer cells which our body, which our immune system have, and through the antibody they generate certain chemical mediators which kills the cells. The same happens with other macrophages which elicit phagocytosis, so-called ADCP. Similar are some of the other systems like CDC, complement dependent cytotoxicity, another mechanism which is driven not by cell but a complement dependent system of the body. So in theory, this ADC, CDC, ADCC, and ADCP are something which is present in our body and is employed by the therapeutic molecule which we want to design killing mainly the oncology targets. Yet another very important aspect of monoclonal antibody is recycling, and this recycling happens through FcRn functions. FcRn is a receptor, neonatal receptor, which is present on cell membrane, and antibodies, IgGs, are characterized to bind to those receptors, those FcRn receptor. This FcRn receptor then internalize it to form endosomes. The pH changes, and because of this pH change, all the other molecule which cannot bind, which cannot remain bound to the FcRn, they are digested by the lysozyme present in this endosome. While since antibody can bind to the FcRn, they are recycled back onto the surface and taken out. Okay. What does this mean? What does this translate into? This translates into the longer half-life of the antibody. Any protein which is given into the body, it gets degraded by this mechanism, largely by this mechanism within the endosome, generating small proteins and ultimately getting rid of from the body. While those proteins which can bind to FcRn, they are recycled back. And this cycle continues until, as I mentioned, the half-life of antibody being 2 to 3 weeks. Now, coming on to therapeutic monoclonal antibodies, getting into just one slide. I'm not a biologist or pharmacologist, but just keeping it very simple. This is a cell membrane, and I'm looking into TNF antibody, monoclonal antibody against TNF. What happens is that in a normal situation, in a pharmacology situation, you have a TNF receptor, TNF alpha, which is a protein, floats and interact with this TNF receptor on the cells, elicit several kinases pathways, and ultimately leading into inflammatory mediators or inflammatory responses. Now, several diseases like rheumatoid arthritis, several types of psoriasis, and other autoimmune diseases are characterized by increased inflammation. And this increased inflammation have been associated in several diseases through TNF alpha system or TNF receptor based system. Now, if we have to develop a therapy to prevent such inflammations, or I should say autoinflammation, is that for small molecule, we can target those kinases. So those, for example, J and K, we can target that and develop a small molecule. The small molecule can enter in the cell and can block this. However, our body have several systems. So if we block that, we will have another chain and ultimately leading into yet another inflammatory response which continues. What can be done with monoclonal antibody is something on the surface of the cell itself. Just to point out here, as compared to small molecules are, of course, they are small, and because of their size, they can enter into the cell, while monoclonal antibodies or large proteins, they cannot enter the cells. So they have to have their targets at least on the surface itself. Now, through the target, it can go into the cell, and I'm going to show you in a short while. but as such, it has to bind, it has to have a target onto the surface itself. Okay. Now, within that, how can we use it? So for TNF, we have already seen that there is a receptor. Actually, there are more than one receptor, so we can see one variant and a second variant. And then there is this TNF, which comes in. It's a trimer. It interacts with TNF receptor and induce some signal, as we saw in the previous slide. Now, from antagonistic point of view, what can we do to block this? We have several options available here, right? The very simple option will be we develop a monoclonal antibody which blocks, which essentially interact with TNF, right? So it interacts with TNF and then it doesn't allow it to interact with TNF receptor. Yeah, that's the option. So here in this case, it's a monoclonal antibody. Another option could be you take this receptor part, just the receptor part which is outside the cell membrane, you kind of graft it on the FC part using the FC part of a monoclonal antibody, so that now the interaction with the TNF and TNF receptor will remain more or less as in the physiological condition. But once again, this receptor is provided or is supplied into the body as a treatment in a higher quantity, such that based on competition, the TNF alpha and the receptor interaction does not take place to elicit any signal transduction. This is how the initial famous molecules against anti-TNF were developed. One of the very initial one using the receptor, TNF receptor, was etanercept. So that's one of those. On the other side, from monoclonal antibody perspective, the first or one of the initial molecule in this anti-TNF category was infliximab, which was a chimeric monoclonal antibody, as you see and also explained previously. Then over the period of time, you will see that using the same mechanism, but humanizing it or more or less developing human antibody, adalimumab, which is Humira, have been developed as well. Yet another strategy is that we use the same variable region, either the fully humanized one or the fully human one or the humanized one, and attach it to the PEG system. All of these have different properties and different um, uh, half-life and different efficacy profile. Again, one more time, targeting the same path. Now, we have seen yet another example in the same line of antagonism will be through a VEGF biology. What happens is that through a VEGF receptor and then cascading through several of the kinases as mentioned here, the signal goes into the body for angiogenesis. Now, angiogenesis is where the blood vessel starts growing. The growth of blood vessel is good in a normal sense, but when the growth of blood vessel is overshooting, it leads to tumor formation, several of them. And it can also affect, if it happens in a very constant area, for example, in the eye, it can lead to macular degeneration and other challenges related to ophthalmologic condition. Again, one more time, the key is related to angiogenesis. Now, one more time, we can generate several different inhibitors, small molecule inhibitors through different pathways here, and those are certainly there available in market as well as discovery. But from biology perspective or biopharmaceutical perspective, the approach we take here is that we try to block this interaction on the cell surface itself. One more time, from antagonistic perspective, you can have an anti-VEGF, so an antibody which is against which binds to the VEGF as such. It can also we can also have an antibody which is against the receptor. So either you capture the protein, you can capture the receptor, or you can have the similar approach as we have seen previously, that we have a monoclonal antibody, a fusion protein, I should say, where we had taken the receptor, VEGF receptor, and grafted it onto the FC part. One more time, the key word here is antagonism. So you try to have blocking of your receptor one way or other. An example using same VEGF pathway is bevacizumab. Uh, an example could be ranibizumab, which is essentially nothing but a part of, or a variable part of bevacizumab. Um, and then there are other molecules as well. Just want to point out what ranibizumab offers over bevacizumab. The small molecule, of course, since it doesn't have its crystallizable region, it doesn't have a longer half-life in the body, but being small, it can cross some of the membranes, especially in the eye. So in the ophthalmological application, it finds much more promising clinical result than bevacizumab. Now, ultimately, we have to come up with what impact those monoclonal antibody brings. And just giving an example here for Avastin, bevacizumab, which we just saw. When we give bevacizumab, so the upper graph, the survival after roughly 2 years is 65% compared to other therapies, non-bevacizumab therapies, which is 42%. So a good increase of close to 25% survival over 2 years is something which is offered. And just to point out, most of the time, monoclonal antibodies being quite expensive, they are not really tried on the patient as a first line. So patients are offered to monoclonal antibody when many of the other simpler treatment have not worked. And in that situation, if we can have an increased survival chance of 25%, it's quite good. Now, let's look at some of the other strategy whereby we can use monoclonal antibody for therapeutic purposes. Here is again one more cell, another receptor, and in this time it's a HER2 receptor, trastuzumab or Herceptin is a monoclonal antibody which binds to it. One more time, it elicits a lot of kinases leading to. So in the normal biology, it leads to transcription and multiplication of the cells. So one more time, this tumor cell increase its population when HER2 receptor gets triggered on. Okay, since this is a tumor cell, of course, from biology perspective, we can block this, which is for sure good. If we can block this, we won't have the tumor cell progression there. But as opposed to the previous conditions, this is a tumor cell. We don't need that, right? And what we can do now, we can employ the secondary function here. As I mentioned, the secondary function which comes through the constant part here, an effector cell. So, for instance, a killer cell can be interacted, can be engaged from the constant part of the monoclonal antibody, as it is depicted in this figure. Once it interacts, the killer cells, the effector cells, release some of the chemicals, chemokines, and that leads to, in this case, for example, perforin. The perforin is released, and this perforin then leads to destruction of the tumor cell. So just consider, in the previous cases, we were just using blockage of the receptor attitude. And here in this case, we are employing not only the blockage, but also destroying the tumor cell using the secondary functions as well. One more time, here the depiction is in a different way. Here it's a depiction of the performance of Herceptin, trastuzumab, where in a HER2 positive, there is an increase overall survival by 18% and this time over much longer period of time. Now, in a similar way, I won't present much more example, but in the similar way as Herceptin employs the secondary function through ADCC, there is another component called CDC, which is non-cell based, but again leads to attacking the target cell, which is a cancer cell, through membrane attack complex. Okay, and through this, it make the cell swell and ultimately burst. So for cancerous cell, for antibody which are designed for oncology system, what do we need? We need to have ADC and CDC to be present in order that those target cells can be destroyed. And through several round of improvement, these ADCCs and CDC function can be even compared to the natural antibody can be even improved, increased. Now, over the period of time, there are additional things which scientists have been doing. One of which is antibody drug conjugate. This is nothing but somehow can be compared with the Trojan horse, where the cell is considered to be the city of Troy. The horse itself is considered to be the ADC complex, and the soldier inside the horse is considered to be the small molecule, which ultimately destroys the city of Troy. And in this case, it destroys the small molecule within the horse, destroy the cell within which these antibodies enter into. How does it work? So antibody, as we have already seen, it's a Y-shaped structure, long half-life compared to other protein, low immunogenicity, human, very close to human, and so on and so forth. So that's antibody. Then we use a small linker, so another chemical moiety, and then we use so-called payload. And the payload is something which is usually very toxic molecule in its pure form, in its natural form. There can be several reasons why we use the strategy. The small molecule here could face a stability problem, can be very toxic if given otherwise, may not have permeability in the cell, etc. So there are several reasons why making ADCs could help overall killing of the cells, or making sure that the payload is delivered into the cell. And how it happens, one more time, if you remember, through the receptor interaction, the antibody gets internalized. Once it is internalized, depending on the enzymes which are present, and this time do remember that this is not FcRn based internalization, this is the receptor based internalization. So in this case, we are actually designing it to interact with the receptor, so-called receptor present on the cancerous cells, getting internalized, degrading, releasing the payload, and this payload ultimately destroying the cell, the cancer cell which takes it. One of the very nice example, one more time, is the same trastuzumab here, Herceptin, or trastuzumab emtansine, where the small molecule maytansine is a very potent microtubulin destructor, I should say. What happens is that this complex ADC interacts with Herceptin, gets internalized one more time, released into the cells, those small molecule, and then this small molecule then interacts in a very specific manner to the microtubule. Now, what's important is that when you give this small molecule, otherwise it will affect all the other cells in the body as well. While if you program it, go only to the target cell, it's only destroying the target cell ultimately. Yeah, that's going into a little bit detail what's happening. It interacts and ultimately through lysosomal degradation, releasing the maytansine, and which inhibits the microtubule assembly, leading to apoptosis. Now, yet another way or advancement, I should say, in monoclonal antibody area is the creation of bispecific antibodies. What are those? In a very simple, there are many of those. So of course, if there is a big chain, if there is a signaling pathway, you can have a bispecific, as the name suggests, bispecific or trispecific, means you have specificity to 2 different targets or 3 different targets, depending on bi or tri. It can be multi-specific as well nowadays. It can target more than one receptor within the same pathway. That's possible. But one of the biggest use, or most promising use, I should say, for bispecific is CD3 based redirection of the immune cells. So CD3 is a receptor on the killer T cells. So this is our human T cells which are there. They possess CD3. This is cancer cell in the patient. T cell is also present in the patient. And if we don't use the bispecific, so if you see bispecific here, this is a depiction of bispecific where you have one arm binding to the tumor antigen and another arm binding to the T cells. If we don't have this second arm, what would happen is that the tumor cell and the antibody, through its secondary effector function, ADC, for example, is engaging the the natural process through NK macrophages, etc., ADCC employment. But this is, yes, this is possible. There are medicines working in this through this route. But if we engage T cells as well, which are much more potent, there are dual role by which the tumor cells is getting eliminated. One is, of course, through ADCC functionality, and second functionality is by the engagement of T cells. So in theory, what you're doing is you're helping the T cell to find tumor cell, and ultimately when they find it, they kill them. I think this is one of the very, very hot field in this area of monoclonal antibody, or antibodies, I should say. There are at least 20-25 different formats which are being investigated in preclinical and clinical trials. This slide here depicts only the different type of, I would say, quite crazy constructs which are there. I'm a CMC person, so from CMC perspective, they are quite fancy. As you see here, the second arm over the other DVD format. There is this SCFV2FC format where in theory you don't have the big part of FC part. The human serum albumin can be programmed to have 2 SCFVs. Then there are bites and darts and nanobodies and so on and so forth. So there are a variety of those. Just one more time to depict how many of those are currently there, being a very, very promising field here. One of which, the CD3 redirector, I should say, got approved, I think 3 to 4 years back, Blincyto from Amgen. And since then, there have been a big increase in interest for bispecific research. As of last year, so this is what I checked last year, I haven't came across anything more recent. But as of last year, there were at least 130 clinical trials which were ongoing, which were registered in US employing bispecific. So very, very important area. Glenmark is fortunate to have their own proprietary, Glenmark/Ignos is having one of the proprietary platform, one of those 20 odd, which I just pointed out. Our proprietary platform is called BEAT, bispecific engagement of antibody based on T cell receptor. Our engineers, what they did is that T cell receptor is a heterodimer where there is alpha and beta subunit. And this heterodimer is formed by the interaction of certain amino acids. I think there are 4 or 5 amino acids which are present in alpha and beta subunits. And what we did is that those amino acids were taken and were carefully planted onto a standard monoclonal antibody. So now we don't get homodimers of those antibodies as in terms of production, but we get heterodimer, and they look something like this. Version 1, which is based on SCFE, or version 2, which is based on 2 standard monoclonal antibodies. Just to point out here, I think we did discuss about a little bit on the dosing part, etc., etc. And for a standard mAb, the dosing here is in the range of approximately few milligram per kilogram. While when we are working towards bispecific, this dose is at least 1,000-fold, roughly in the range of 1,000-fold lower than a standard monoclonal antibodies. Okay, now apart from the standard use, there is other use which is by radio labeling. So you can have a standard mAb, again through using chemistry and so on. You can attach a radioactive complex onto your monoclonal antibody. What are you using here? Your monoclonal antibody is a very specific, very targeted one. So through its specificity, it will go and interact with the tumor cell, and through its radioactivity part, which you have attached through a chemical linker, based on gamma or beta radiation, it will kill the cells which are around it. So in this case, one more time, what we are doing, a so-called magic bullet, which was talked earlier. In theory, now radioactivity using this, in this case for Bexar, is in the very confined region, the region where you have the target available. This example for Bexar is using iodine 131 and is an anti-CD20 antibody. Now, yet another use, not very widely, I would say, used, but still there is a approved product there. We know that our heartbeats are governed by a sodium potassium pump, and digitalis, which is a glycoside, is something which can influence, which can regulate the sodium potassium pump. It can control the sodium potassium pump. But if wrong dose of digitalis is given, then we are in trouble. It's a similar situation as we have with insulin versus glucagon. So if a higher amount of insulin is given, you need to have glucagon to compensate it. In this manner, the way it is being used is that antibody against digitalis, which is a polysaccharide, have been generated. And as I mentioned, for polysaccharides, IgG2 are one of the best ones. So a digoxin specific antibody, which is called DigiBind, have been generated and is used as an antidote. One more time, I mean, digoxin toxicity is a life-threatening one, and that molecule DigiBind is a savior in this case. This slide here, I'm just trying to summarize what are the different things monoclonal antibody can do just onto the tumor cells. So there are several different applications which are driven by standard mAb. So just the plain mAb, I should say. Then there are functionality which are related to conjugated mAbs, whether it's ADC or through radio immunology. There are then bispecifics, which we discussed. The last thing which is there is something called CAR-T or cell therapy, which I won't say completely is driven by mAb, but I will strongly say this is truly inspired by monoclonal antibody as well. So in theory, what we are doing is that using the same specificity which antibody offers, you program the cells, the patient's own cell, to produce those antibody onto the cell surface and use them as therapy. So it's really inspired from the specificity which a monoclonal antibody offers. So this is kind of the therapeutic use of monoclonal antibodies. Just quickly, before I finish, there are a few other applications as well. The first in diagnosis, I'm sure most of you must have seen a pregnancy test strip or a malaria testing strip. What does it have? It has a simple sample pad where we add blood or urine, and then there are 2 or 3 lines, so-called, which is normally one is a control line just to make sure that everything has worked, and then there are 1 or 2 test lines. How does it work typically? So in theory, you have the analyte, which is, as I mentioned, sample from blood or urine. Then there is a complex of antibodies, which, if depending on the positivity, depending on the affinity interaction with the analyte, it binds, and then because of capillary flow, it flows into the direction. And then there are the second antibodies which are present on through the test line, and then the control antibodies which are present in the control line. And depending on what we have to test, for positive, there will be interaction, or the second part of the sample will bind to this positive test line, while the control will simply be performing a check that flow and everything has really worked. COVID is hot, and there have been this test which have been employing exactly the same thing one more time, really driven by IgGs. In this case, the purpose is to detect the presence of IgGs and IgMs. And in this case, there are 3 lines which are for control. The presence of IgGs and the presence of IgM in the blood of the treated patient. Finally, just one slide on the use of antibody in production. So if we know we have gone through this cycle, and once you have a very specific antibody which is available, it's a very powerful tool. You can immobilize them on a column and then can use it for purification of your so-called previous protein, which was antigen at one point, to purify it over and over and over again. So that's yet another, it's not I would say not sheerly a therapeutic application, but something which is very, very commonly used into biopharmaceutical. Now, I would conclude with following points. The key point for monoclonal antibody is that they are highly specific and they have a long half-life. They are redefining disease management, especially in oncology and autoimmune diseases. There are several of them, and now getting more and more advanced. Radio-label antibodies and diagnostic antibodies are also helping healthcare system and actually advancing the healthcare system towards more and more personalized medicine. Now, whenever a clinical trial is being initiated, patient stratification is a very, very a standard operation, especially in many of the oncology area. And then advanced modalities like bi-tri, multi-specific, as well as cell therapy, which are really inspired by antibody-based therapies, are something to be looked at for future. And with this, I would like to thank you and be happy to take discussions and questions. Hello.
Dr. Pethe? Hello, Dr. Pethe, are you around?
Yes, yes.
Yeah.
Yeah. Sorry, I was on mute. Thank you, Dr. Sachin, for delivering us such a wonderful and informative session. Just to summarize your presentation, you started with discussion on immunoglobulins and their types, the role of FcRn, VEGF biology, anti-VEGF. You explain the mechanism of few molecules like bevacizumab, trastuzumab, herceptin. You thoroughly discuss about the antibody drug conjugates. Then you highlighted the recent developments like bispecific and multispecific antibodies and their application, and the various molecules which are under clinical trials. You spoke about the monoclonal antibodies as antidotes, as diagnostics tool, and in purification of proteins. Very nicely covered the various applications of monoclonal antibodies. So thank you very much for your lucid presentation. I will take few questions. Let me see the chat box. Yeah, the first question is from another Sachin. What are the ethical issues of monoclonal antibodies?
I mean, I'm not sure. So let me put it this way. I think from an ethical perspective, the issues from development perspective, to my understanding, are no different than for a small molecule or any other drug from development perspective. And actually, as Suneet mentioned previously, there are many studies which are not required. So like they're considered to be more safer in terms of not eliciting any cardiac based response or liver based toxicity studies. So in theory, they are considered to be a little safer in certain aspects, but then there are some other aspects like immunogenicity, where a more thorough evaluation is supposed to be performed. One more time, I think from ethics perspective, so ethics come into play when you're trying something and want to make use of it. And to my understanding, the chances of success with biologics as compared to small molecule, I will say, will be much more. So I mean, we know all this funnel which require 10,000 molecule, 10,000 small molecule to come to one drug ultimately. I would say when it comes to biologics, this 10,000 molecule will be much less. It may be, I don't have a number, but it could be a few hundred of those to get into the last one. So from ethical point of view, it's actually a favorable situation. You don't try a lot of molecule which won't work ultimately.
Thank you, Sachin. One more question for you. And the question is from Mr. Purav Shah. What could be the upcoming challenges in formulating a drug product with modified bispecific monoclonal antibody, especially with respect to stability when compared to the conventional monoclonal antibodies drug product development?
Absolutely. So the biggest challenge is coming from its biggest merit actually. The MAB, a standard MAB is, as I mentioned, few milligram per kilogram is the dose. For bispecific, this is few microgram per kilogram. That means what we are working at is roughly 1,000-fold lower and lower concentration of protein through a very different array of challenges. So that is something which have to be looked at very carefully. Absorption prevention and some of the challenges related to excipient protein ratios. And then on top of that, there are challenges by virtue of their structure itself. As you have seen, the structures of bispecific are quite different. There are roughly 65 standard monoclonal antibody which are in market. So a lot is known from the personality, from the stability perspective for a standard MAB. But bispecific are relatively new and their structures are very different. So we cannot leverage on from that information that we have the possibility with IgGs. So those are, I would say, two prime challenges I will focus, and I mean, we focus on to make sure that it's well taken care of, apart from all the other things.
Yeah, thank you, Dr. Sachin, for your answer. One more question, and I think this is the last question for you. Can bispecific be used for diagnosing more than one marker simultaneously?
If I to give a very short answer, then in theory, yes. But it's important to make sure, because the bispecific can bind to two different signal molecules. But then how will that transform it into, let's say a color reaction? The, the two different binding have to lead into two different, let's say, colors in a very simple term, which is gonna be some challenge, I will say. So it can be explored, but I don't think, considering all the different challenges which are there with bispecific from the stability perspective and so on, as well as from operational perspective, that you need to have two different pathways or two different colored signal, one for the one arm and second color for the second arm. It requires some working still.
Yeah, thank you, Dr. Sachin. Here we conclude the last session for the day. So, as a token of appreciation and on behalf of CRS-IC, I would like to present a virtual certificate to you, Dr. Sachin. The certificate is there on the screen. Please accept this on behalf of the CRS-IC. And as it is mentioned on the certificate to go green to save the planet, it's not only written on the screen, but actually we are trying to save the planet by planting a tree. And the first time the CRS-IC has taken the initiative, very nice initiative, and we will be planting a tree at Indira College of Pharmacy with your name to commemorate your contribution in the field of CRS. So thank you, Dr. Sachin, for taking out time from your busy schedule and honoring us with your presence. Thank you very much.
There is something which I would like to add. I mean, this is really a bond which you are making for us. This is, of course, an honor for us, but you are making a bond for us with that tree, which is gonna be in Pune. And given a chance, I would certainly like to water that tree at one point.
Wow, that's
You are
good.
most welcome, Dr. Sachin, and Dr. Kulkarni.
Yeah. Yes, Dr. Sachin, you are most
Yeah, she
welcome
will
to
host
our
you.
college. Yeah, you are most welcome
Thank
to
you.
our college, and we'll very soon be planting all these three trees in the names of the three speakers that we had today, and we will surely send you the photograph while we are at least planting the trees. And when you physically come to India, I would definitely urge you to kindly visit our campus and see the tree grown.
It's a joint responsibility, and I will certainly do that.
Yeah. Yeah, thanks a lot. So now we've come to an end of this knowledge packed workshop. I would request Dr. Vandana Patravare to kindly propose the vote of thanks. Dr. Vandana is a professor of pharmaceutics at Institute of Chemical Technology, Mumbai. She's been associated with CRS-IC right since its inception in 1994. And currently, she's working as a vice president of the executive committee. Ma'am, I request you to please propose the vote of thanks.
Thank you, Madhur. So on behalf of Control Release Society Indian chapter, it's my proud privilege to propose vote of thanks. Our sincere thanks to Mr. Ajit Singh, Chairman ACG Group, for taking time out of his busy schedule to be with us this evening, in fact, to inaugurate the revamped CRS-IC website and share pearls of wisdom with us. In fact, I would say that his support means a lot to us, and he has been associated with us since inception. We do look forward to his valuable suggestions always, as well as the support for our future programs too. Our gratitude unlimited to our speakers of the day, Dr. Rajeshwari, Dr. Suneet Shukla, as well as Dr. Sachin Dubey. In fact, Dr. Sachin mentioned hats off to participants. I would say hats off to you speakers to spare your valuable time on weekend, and that also at odd hours to be with us at CRS-IC. No, we really sincerely thank you for being true teachers. And just like, a compass activates a magnet, you have actually activated our interest, the curiosity, the knowledge, wisdom in all the participants in this frontier area of monoclonal antibodies. I'm sure this is just the beginning. There'll be many more such sessions as some participants have requested, and we'll see that we can do the needful. Thanks are also due to our coordinators, the session coordinators, Dr. Sujata, Dr. John D'Souza, as well as Dr. Anil Pethe for smooth conduct of this program. And special thanks, of course, to Dr. Anagha Joshi from Indira College of Pharmacy for being graciously accepting our request to plant the saplings on campus on behalf of CRS-IC. And definitely it's a bond which has got formed not only across borders, but also within the country. CRS-IC also wishes to applaud efforts of Tarun Soni, Director, Novel Innovative Concepts Interactive Limited. In fact, for designing this particular website, wonderful website, which is very, very user-friendly, and his inputs, tips, suggestions have been so fruitful, and we wholeheartedly appreciate them. Success of any program depends on vision of the leader and support from the team members. And we need to wish, in fact, we wish to thank Dr. Parizat El-Chidana, our visionary leader and president of Controllerly Society Indian chapter, as well as each and every member of EC, Professor Udupa, Dr. Madhur Kulkarni, Dr. John D'Souza, Dr. Anil Pethe, Dr. Yogeshwar Bachao, Dr. Sujata Savarkar, Dr. Pranav Shah. Believe me, each one of them really have taken efforts to see that this program is a huge success. Special thanks also to Purav Shah, our social media coordinator. And in fact, I can see our past president, Dr. Anisha Pargal, who's been with us right from beginning of the program. Thanks, Anisha, for being there and for supporting us in all the ways possible. Just for information of viewers, each and every EC member gives his or her time purely on volunteering basis for the benefit of society at large, without anticipating anything in return. And that's a trend which was set by our founder president, Professor Balla. And I'm sure we'll take on that challenge and move ahead and do our best in days to come. And finally, dear participants, the CRS-IC is for you. Do let us have your suggestions, feedbacks for future programs, for future workshops, if any, and we'll strive to do our best. Thanking each and every one of you who's present till so late from bottom of my heart on behalf of CRS-IC. Stay safe, take care, namaste.
Thank you, ma'am.
Thank you.
Dr.
Thank
Madhu,
you.
you are on mute.
Thanks a lot, ma'am, for this. And we cannot conclude the session without thanking you enough, especially because today, while Dr. John was coordinating the session and I was a backup, both of us lost internet connectivity at the most, I mean, what I can say, the most crucial time. But Dr. Vandana Patravale, Vandana madam, actually took over so swiftly and did the needful without even creating or without even making others understand that there was something wrong actually there. And I would also like to thank Mr. Tarun, because he also did the presentation of the certificate also at the most crucial and the right time. So thanks a lot. Thanks a lot to all of you. Thanks a lot to the participants. And just to let you know that the feedback link has been put in the chat box. So I request all the participants to kindly fill the feedback after leaving, or I mean, just click the link and start filling the feedback. If you have any problem accessing the link has also been posted on our CRS-IC website. So do visit the website www.crsic.org, and you can find the feedback link there as well. On behalf of Team CRS-IC, I wish you a very good night and Shubharatri. Thank you.
Thank you.
Thank you, Dr. Madhur. If you permit, then I will close the meeting.
Yeah, I think we can. I think we can keep it on for a while, because if participants want to copy the link and fill the feedback form. Thanks a lot, Tarun. Oh, you can keep your video on actually, because I think we have never seen you. At least I have never seen you before.
Oh, you've not seen Tarun?
No, I mean, he's always
Okay.
been keeping his video switched off. So this is the first time
SPDS meet is when we actually saw him first. Thanks, Tarun, for being a wonderful support.